Well, uh, thanks everyone for joining, and I am really happy to introduce Dr. Nina Zhou. Hopefully, I did a better job pronouncing it than I have in the past. Um, in any case, Nina has been working with us here in San Diego for over a year now, I believe, and she is a postdoc in the Dorestein Lab at the School of Pharmacy and Pharmaceutical Sciences, and she focuses on chemical exposures and how they're related to health outcomes. And she does this using metabolomics data and computational tools. And a little anecdote, I listened to a lot of podcasts. I listened to one a couple of weeks ago about a toxin from car tires, from the rubber of car tires that's toxic to salmon. And then I read that Nina actually did that research and discovered this. So Nina, I hope I connected those two things correctly, but I'll let you go ahead and launch into your presentation.
Thanks, Ron, for the introduction and also mentioning the higher chemical and salmon health work. I would love to share that with you today because that's such an interesting study. But like, uh, unfortunately, that's not the focus of the talk today. But I hope the topic today will be even more exciting and more encouraging for the audience here, where I'm going to share you a resource we recently developed that can help you screen drug and drug metabolites using non-targeted metabolomics. A couple of weeks ago, we hear a wonderful talk from the Johns Hopkins University about using mass spec to detect tenofovir and imtripsitidine across the blood-brain barrier. And today, I'm hoping to basically do similar things as that, but then we'll be expanding the capacity to hundreds of thousands of drug detection at the same time. So a brief introduction, as Ron mentioned, I'm interested in chemical exposure and their effects in the human health. And we all know that food and the chemicals in the environment and the microbe, they all have tremendous effect on the health status of our body. But among all this different chemical exposure, we pay special attention to drugs because those chemicals were designed to be bioactive and they can have a profound impact on our health, not only the whole system, but also the gut microbiome. And because of the focus on drug exposure, I get a lot of requests about drug analysis in my daily life. So one of my collaborators, she was interested in infant health, and she brings thousands of human milk sample to me and asked me, Nina, can you use your magic on targeted metabolomics and tell me what are all the antidepressants you saw in this human milk sample? So to just give you a little bit of background of what is non-targeted metabolomics in case the audience is not familiar, basically I use the instrument called mass spectrometry, which can help me detect all the chemicals in milk sample, basically, like the gray dots you saw on the instrument and the milk. So to identify, to know what are those chemicals, the instrument can help me produce tandem mass spectra, which you could basically think of it as chemical fingerprints of all the chemicals we saw in the human milk sample. So to translate these mass spectrometry signals to chemicals that we actually know, the way we perform such analysis is by comparing those tandem mass spectra to a library of spectra of known compounds. So for example, here you would recognize that the second mass spectra actually looks the same as the spectra of caffeine as shown here on the left. So basically through this strategy, we will start to know that this human milk sample has caffeine in it. This mom potentially has consumed caffeine, and then it transport to their breast milk sample. So it sounds easy and simple enough, but that's actually not how the story goes. So when I compare the mass spectrometry signal against the spectral library, it's not only a simple name like caffeine, but I can get names like the ones here, which including super complex chemical name that no one can understand, as well as a couple of drug name that's really unfamiliar to me. So I came from a chemistry background. I don't know a lot about like pharmacology. So to really understand what are all the antidepressants I detected in the human milk sample, I basically need to Google or nowadays talk with ChatGPT for a really long time. And finally, I will be able to get a short list of antidepressants from all the chemicals that I detected. But the story never ends here. Another collaborator came to me, and this time he was interested in human gut microbiome. So he wants to know all the antidepressants I detected in the fecal samples they provided to me. So I basically did the same thing. I used my magic on targeted metabolomics, and I was able to detect thousands of chemicals in this fecal sample. To identify all the antibiotics out of the chemical detection, I basically spent another really long period of time Google about these chemical names one by one, and finally I was able to shortlist a bunch of antibiotics I saw in the fecal samples. When the third collaborator came to me, and this time he was interested in what are all the illicit drugs I can detect in the wastewater sample, I was like, hold up. I love science and I love metabolomics, but I don't want to spend the entire of my career keep Googling about chemicals. So instead, all this request about drug analysis really motivated me to think, can I actually develop a one-stop shop for drug analysis? So the idea is that I want to compile a spectral library that's specifically for drugs. And more importantly, I want to connect all the entries in my spectral library to the metadata of the drug. What disease do they treat? What is their mechanism of action? So on and so forth. The hope is that with such a resource, no matter what sample I receive in the filter, no matter if it's breast milk, it's water, or it's fecal sample, I can just run the samples on the instrument, compare them against my spectral library, and using the metadata in 1 second, I will be able to tell you all the antibiotics I saw, all the antidepressants I saw, so on and so forth. So I started really working on it, try to construct such a resource. So the first step would be to acquire reference mass spectrometry spectra for the drugs. So to achieve that, we utilize the GMPS library, which is a publicly available collection of all the mass spectrometry reference we have. So the reason we use the GMPS library because it's already composite most of the common open access mass spectral resource such as mass bank and the FDA mass spectrometry library. So the way we search for drugs from the GMPS spectral library is through a partial name search. So I accumulated all the names of drugs from this 4 knowledge base I shown on the upper right corner. And then I search for my spectra. I search for chemical in the GMPS library whose name contains the full name of the drug. So for example, when I search for venlafaxine, I capture not only venlafaxine, but also desvenlafaxine, which is a metabolite of this drug. So through this effort, I was able to collect the reference spectra for not only the drugs, but also the drug metabolites. And in total, I was able to collect about 76,000 drug reference spectra from the GMPS library. And this represents about 2,000 drugs as well as metabolites for about 100 of them. To increase the coverage of our library, of course, it's never a bad idea to purchase drug standards and then generate the novel spectra from them. So to do that, we collaborate with scientists from Czech Republic, and Karina, who is a hero of this project. She got access to reference standards about 20,000 chemical. Among them, about 5,000 of them is drugs. So we were able to utilizing this resource and collected about 30,000 reference spectra that belongs to 4,000 drugs. In addition to expand the coverage of the metabolite reference library, we collected with Dr. Libin Xu at the University of Washington, who has cultured about 2,000 drugs with a human liver in vitro system. And through that, we were able to get a reference spectra for about 470 metabolites of the drugs. So combining all this resource, the GMPS library, the new reference standard, as well as the metabolism assay, in total, we were able to accumulate about 100,000 reference spectra for around 5,000 of drugs. And about 470 of those drugs have corresponding reference spectra for their metabolites. So as you can see from this number, this is already a great resource, and we have spent a lot of time trying to collect not only the reference spectra for the drugs, but also for the drug metabolites. However, you could see that we still only have about 10% of the metabolites for all of the drug. The coverage for drug metabolites was still pretty low in our resource. So we want to collect more reference spectra for drug metabolites, and how are we going to do that? Well, we turned to public metabolomics data sets that hosted in the GMPS repository. So for those of you not familiar, GMPS is not only a reference spectra library, it's also a platform that allows you to store your metabolomics data and perform data analysis. And all the data you uploaded onto GMPS will be available to the entire metabolomics community to look at them later. So when this project happens, GMPS already hosted about 27 studies, GMPS already hosted 2,700 studies, 2,700 projects, and that represents about 1.2 billion of MS2 spectra. The resources continue to growing, and right now we already expand this number to about 5,000 studies. So our hypothesis is that there might be drug metabolites that has been already detected in these public metabolomics data. We just need to use some computational tools to find them, to identify them, and so that we can use those drug metabolites to help the drug detection for filter studies. So the way we find drug metabolites from these public metabolomics data is to using an algorithm called modified cosine similarity. For those of you not familiar, this is basically a strategy to look for similar fragmentation pattern between two chemicals. So for example, as you can see between the structure of venlafaxine and this venlafaxine their structure was different by a methyl group, and their MS2 spectra show this consistent pattern of 14 Dalton changes. So modified cosine similarity basically help us to recognize such consistent changes in the MS2 spectra, and then it help us to connect the drug and the drug metabolites together. So we use this strategy and we search across the entire repository for MS2 spectra that looks similar to the spectra of a drug. And through that effort, we were able to capture about 3,000 unique spectra, which represents 3,000 unique unique new structures that belongs to the metabolites or analogs for about 600 drugs. So to differentiate these computationally derived drug metabolites to the ones we collected from reference standards, we gave these signals the name of drug analogs. So in the future presentation, I'm going to refer to them as the drug metabolites we collected from the reference databases as drug metabolites, and for this computationally derived drug metabolites, we will refer to them as drug analogs. So now we have a spectral library for drug metabolites, and drug analogs. As I promised to you at the beginning of this presentation, when you use our drug library, you will never need to Google about drug again. So to achieve that, another component we want to add it to this drug library was a drug metadata attached to all the reference spectra. So originally, I developed this tree-based drug metadata based on the information I collected from these knowledge base, these drug knowledge base listed at the bottom of the screen. However, when I show my resource to a pharmacist in our department, Dr. Kine, she noticed a lot of inconsistency in these resources, and she was so mad at me. She was like, Nina, how can you introduce to the audience a resource as chaotic as this? So she, in the end, took the heroic effort to go through all this information and manually curated them one by one. So basically, I want to give a big shout out to Kine here, but also if you find any mistake in the information provided by this resource, contact her, it's not my problem. I'm just kidding. But after we have this information on the therapeutic area, mechanism of action on the drugs, another caveat we noticed with resource is that it also contains a lot of molecules that could also come from food or endogenous sources. So for example, our current drug library include bile acids, which could be used as a drug to treat liver disease. But of course, the major source for this chemical would be our endogenous metabolism. The current drug library also contains hormone chemical. They could be produced endogenously, but they could also be used as a drug to treat endocrine disease. The drug library even contains food additive because these food sweetener could be used as laxatives. So these chemicals, although they could be used as a drug, their exposure indication would be very different compared to the chemicals that can only be used as a medication. So to solve this problem, we curated another layer of the metadata for the drugs included in our library, which categorize them based on their exposure sources, including medical source, or if the chemicals could come from personal care products or industrial sources or endogenous or food molecule. This layer of metadata was again curated through manual curation by me and also to students. So a big shout out to them as well. So now we have developed a resource that contains reference spectra about 5,000 drugs together with their metabolites, as well as this structured metadata for all the entries in our drug library. So the next step, I'm sure you will be thinking about this throughout my presentation, is how reliable is this drug library? So to test that, we first utilize the plasma sample we could get from the public repository. The first one is on HIV patients, and the second one is on COVID patients to compare their drug exposure profile. So the bar plot here shows the most commonly detected drug in these two cohorts, and we could see that in the people with HIV, most of the medication we observed were indeed antiretroviral medications. So it agrees with the drug usage for people with HIV. However, for the COVID, we saw antibiotics, we saw drugs used to treat flu, we also saw corticosteroids. These plasma of the COVID, people infected with COVID was collected at the beginning of the pandemic, and people were sent to the hospital because they have really severe symptoms. Basically, the drug detection was telling us that at that time, early COVID, during the early stage of the COVID pandemic, we really have no idea how to treat this disease, and we were using all kinds of different drug cocktails on these patients. The second small test we did was to compare the drug exposure profile between an elderly and a young healthy cohort. And this time, we compared the brain samples from Alzheimer's patients versus the human milk sample we collected from moms. So I categorized the drug based on the disease area accumulated in the drug library resource. And we could see that, first of all, the human milk sample have significantly less number of medication detected compared with the brain sample. And when we look into some of the most significantly different categories, such as the neurology medication, antihypertension medication, and the hematology medication, we could see that even within the neurology category, the brain sample from the Alzheimer's patients was the drug that detected there was used to treat depression, Alzheimer's disease, and schizophrenia, which was common disease experienced by this cohort, versus the neurology medication observed in the mom mostly include antidepressants, pain relief drug, and epilepsy drug, which was also reflect the drug usage for this young cohort. So hopefully I was able to show you that the drug library could sensitively reveal the drug exposure in different disease cohort, and that gives you a reliable readout of the drugs in different disease categories. So at later stage of this project, we were really honored to have received about 3,000 fecal samples from the HNRC cohort. And because previously we showed that the drug library was very sensitive at doing the drug exposure readout from people with HIV, so we decided to do a deep dive on our analysis to these fecal samples. So again, we received more than 200 fecal samples from people with HIV, and we also include control samples in this cohort, which include about 100 samples from people without HIV. Using the drug library, we observed in total 167 medications in this cohort, and the most popular drug category include neurology drug, infectious disease drug, as well as cardiology drug. So the detection of neurology drug, the detection of infectious disease drug makes a lot of sense because these are basically antiretroviral used to control the HIV infection. And the detection of the neurology drug and cardiology drug was most likely due to the comorbidity commonly observed in people infected with HIV. What's interesting is that when we look deeper into these drug detection, we notice that about half of those drugs was actually detected not only as the drug itself, but also together with the metabolites or analog library we curated earlier. So the dark blue color highlighted the drugs observed together with their metabolites and analogs, and about 33 of the drugs we observed in this cohort was together with these metabolites. So we want to know, especially for the computationally derived drug analogs, we want to know whether these signals was relevant, whether these signals was real drug metabolites rather than some random noise, right? So here, I showed the detection of the drug, the Ranavir, which is a common antiretroviral. We show the detection of this drug as well as all the analogs, all the drug analogs I observed in these samples. So here is the heat map. Each row in the heat map was one of my detection, and each column in this heat map represents one fecal samples included in this whole cohort. The top row was the drug itself, and the bottom row was all the computationally analog I derived in previous step. And we could see that the observation of these analogs in these fecal samples agrees very well with the observation of the parent drug. Basically, the observation of these drug analog corresponds to the exposure for this parent drug rather than some random noise that we derived during the computationally mining process. So beyond the Ranavir, when we expand such analysis to other drugs, we saw most likely the similar signals. So not only that the Ranavir analog was observed together with the parent drug, but we could see on this heat map that for most of the drugs we saw in these samples, for most of the drugs we saw in these samples, the corresponding analog, their occurrence agrees well with the parent drug. So again, it's showing that the computationally derived analogs was not some random noise, but they could be signals that's relevant to the exposure of this drug. In another word, they should be real metabolites for these drugs. Then the next question is, what are these analogs? Where do they come from? So to answer this question, we selected 13 drugs that we observed in these samples, and we cultured them with a synthetic community of the human gut microbiome. The purpose for this experiment is really to see if gut microbiome metabolism could be one potential source for these different drug analogs. And we selected the gut microbiome community here because these drugs was observed in the fecal sample, so we thought the microbiome was a relevant metabolism pathway that we want to target it here. So here are some examples of the drug analogs we observed together in the fecal sample as well as in the microbial culture. You don't need to understand the technical detail, but basically the purple color was the signal we observed in the fecal sample from this cohort, and the red signal were the red color were the signal we observed during the microbial incubation. So basically, these plots were showing that for these drug analogs, we observed they were the safe chemical that was observed both in the fecal sample and during the microbial incubation. Basically, through these experiments, we want to show that yes, the drug analog are real signal, and some of them were coming from gut microbiome metabolism. So now, hopefully, I have convinced you that the drug library is a useful resource and it can provide reliable drug readouts from a diverse class of sample. And I guess the last question I want to address here today is, why do we actually need to measure the drugs? When we built this clinical cohorts, we collected their drug usage, drug exposure profiles through their clinical records, right? Why do we want to bother to analyze this drug using mass spectrometry and use this drug library to really detect their drug rather than rely on the clinical record? So I'm going to show you the value of the drug library utilizing our analysis on a class of microbial metabolites called N-acyllipids. I'm very interested in this class of molecule because they were really important signaling molecules provided by the gut. And also one postdoc in our group, Dr. Helena, she previously observed that some of these important signaling molecules, some of these acyllipids, their level was higher in people with HIV compared with people without HIV. Of course, when she saw these results, she wants to understand whether that change in the signaling molecule was caused by the HIV infection or it was caused by the antiretroviral usage for these people. So what she did was she utilized the clinical metadata provided by HNRC to us, which categorized the drug usage of these participants based on the number of drugs they were currently using. So from ARV naive, which means they never use ARV, no ARV means they currently do not use ARV, or current ARV number lower or higher than 3 drugs. So what Helena observed is that based on these category of the participants, she did not observe any significant difference between the level of these acyllipids. So basically, the conclusion at that time was, well, maybe drugs doesn't have an effect on these microbial signaling molecules. It's because of the HIV infection that we observe changes in these signaling molecules in people with HIV. But of course, I'm not convinced by this result. I believe drug has a really important effect, so I decided to look into this question deeper. So I analyzed the drug exposure profile I analyzed the drug exposure profile, especially the HIV medication in these participants. And here is a heat map again, like the one you saw earlier. Each row in the heat map is one antiretroviral I observed in these participants, and each column is one participant, with one sample, one participant. And here at the bottom, I have a bar which noted the HIV status of these participants. Blue color means HIV positive, and the yellow color means HIV negative. And first of all, we could see that the drug exposure was very different between people with and people without HIV, which makes a lot of sense because because people with HIV, we saw abundant signals for HIV medication versus the signals was really rare in people without HIV. And when we look deeper into the drug exposure profile, we notice that although the people with HIV all use different, all have drug detection, their drug usage profile was actually quite different. So they form distinct clusters here on the left part of the plot. So for example, we saw lamivudine, abacavir, and the dolutegravir. These 3 drugs often show up together in this cluster of participants here. And indeed, these drugs was commonly used as a cocktail treatment to stop the HIV infection from multiple mechanisms of disease. So based on my observation that the people with HIV use different group of drugs, I performed a cluster analysis, and at this time I took out the sample from people with HIV only, and I clustered them based on their different group of drug exposure profile. So you could see that these samples were separated into 4 groups with very different drug usage here. So after I performed such clustering based on their HIV drug usage, I noticed that this time the acyllipids, the microbial signaling molecule, showed a significant difference among these different drug usage groups. And I want to emphasize here that this analysis was performed only on people with HIV. It means that we have already controlled the infection status to this virus, and we only consider the effects of drug exposure here. So basically, it is showing that not only the HIV infection, but also the drug exposure that is causing changes to these microbial signaling molecule. And we also noticed that it's not only the one molecule that was observed previously, but it's a group of this signaling molecule that showed significant difference between these people with HIV, but using different class of drugs. So with this example, I really want to demonstrate to you that using untargeted metabolomics and using the drug readout with our drug library, we could discover biological patterns that could be ignored or missed if we only rely on the clinical metadata provided to us. So with that, I hope I was able to introduce to you a resource we recently developed that allows you to perform one-stop shop analysis for the drugs. We construct a resource that contains not only reference spectra for drugs, but also metadata connected to this reference spectra to help us easily interpret and understand these drug detections. We show that a lot of the drug analogs we derived from mining public repositories could be likely microbial metabolites for this drug. We also show that drug exposure was connected to microbial signaling molecules that was observed in the fecal sample from the HNRC cohort. Before I let you go, I want to give you one last quick and simple example of how fast the drug library functions. So Shirley, who is actually the PI at the beginning of the presentation that asked me about antidepressants in human milk sample. So one day she approached me at about 11:30 a.m., which was right before my lunchtime. She asked me, Nina, do you have preliminary data on the 2,500 breast milk sample about what other SSRI you could see in them? So I was at lunch with my colleagues at that time. So I finished my eating and then come back to my office. And at 12:22, I was able to give her the number. Basically, within an hour, we were able to perform the drug readout in about 2,500 breast milk samples. So Shirley was very happy at the speed that I functioned. She was like, awesome, thank you, Nina. But I really want to say that it's not me working really hard, that's really awesome, but it's because I have developed the drug library that allows me to perform such analysis in such a fast speed. So with that said, here's my acknowledgement slides. I'd like to thank all the members in the Dorothy Lab that has helped me through this project, as well as all our funding agencies. The work has been organized into a preprint here. So if you want to try our metabolomics resource, feel free to scan the QR code. But of course, you could definitely email me, and then we could start to think about how we can work together and try to understand drugs in your sample and what are their impacts to your to the outcome that you care about. So thank you all for listening, and I'm ready to take any questions you have.
Thank you, Nina. That was wonderful. You did what a lot of speakers don't do, which is you left a whole bunch of time for questions. A little bit risky, but let's see what the questions are. Maybe.
Should I modulate, or Ron, you could pick the audience and ask a question?
I'm
I'll
not
let you modulate. I'll let you modulate.
Yeah, I'm not seeing anything in the chat. There are quite a few people in the audience. Maybe you stunned them.
I have my hand raised.
Oh, I'm sorry. I can't. My screen is so full of people, I can't see them.
So anyway, so it was okay if I go, Ron?
Sure, sure.
So I've got 3 questions, but Ron will stop me if I go on too long, or you can, Dr. Zhou. So one, when you were talking about corticosteroids, I was thinking, you know, a lot of the medications that we prescribe are designed to mimic or actually be endogenous molecules. So insulin, steroids, opiates, hormones. So can the library distinguish endogenous molecules from exogenous molecules if they're exactly the same or very similar? Oops, sorry, I did that.
That's actually a great question. That's also something I have been thinking about. So right now, let me show this slide actually. Right now, if this chemical has an endogenous source, like you said, if it's an insulin or if it's a hormone, we tell you that this chemical has endogenous source. And during our downstream analysis, we typically exclude those chemicals because we don't know if that was prescribed as a medication or if that's produced by your body. We provide this information, but then it's up to the user how they will utilize, like whether they will consider these drugs as drugs or whether they will consider them as endogenous molecule and exclude them from the drug folder. But one thing I have been thinking actually is whether we could tell apart if it's a molecule was produced endogenously or is used as a medication. Maybe we could use concentration or basically signal intensity to differentiate prescribed using of that molecule versus endogenously produced molecule, but we haven't really looked deep into that yet. Right now, in our downstream analysis, we just don't consider the molecule with endogenous source.
So the medications are usually formulated with excipients, chemicals that help to make them into the pill and to be absorbed after breakdown by gastric acid and so on. So perhaps we could look at some of these medications and see what excipients are commonly included in the oral medications and see if you can detect those or not. And maybe that would help us distinguish whether it could be, whether it supports that it's exogenous rather than endogenous. But anyway, that's only one. Two is, we've talked about the histamine finding, and we found that very intriguing. And when Peter and you asked us about it, I focused on the fact that people with HIV are more likely than people without HIV to have allergic medical conditions. So including, things like asthma and allergic rhinitis. And so, and Ron, said it's also a neurotransmitter, and he was absolutely right. So I think those are very intriguing findings, but as a result of people with HIV being more likely to have allergic conditions, they're also on antihistamines. And so it would be interesting to also look to see if what the relationship is between antihistamine use, if there are metabolites for those, and the histamine findings that you have.
That's a very cool idea. We will definitely look into that. I do believe we saw a lot of antihistamine in this sample, so that would be something really interesting to look at.
All right, and I don't see anyone
totally
else's.
Agree.
I don't see anyone else's hand raised. So my third and final.
Except for mine is raised.
Oh, well, I didn't see you, Dr.
But you go ahead. You go ahead. You might ask the question that I was going to ask.
Well, so Ron probably has thought of this, and I don't know if he's talked about it with your group or not. But people with HIV are less likely to respond to antidepressants, and so the last thing that you were doing made me think that typically we have to assess that based on something like Beck Depression Inventory, but it would be really cool to look at specimens before and after initiation of antidepressant therapy to across people to see what the inter-individual differences are in response of serotonin and tryptophan and norepinephrine and other things that we don't commonly measure in antidepressant therapy, to see if we can identify people who are more or less likely to respond to different antidepressants based on metabolomics. So,
do we have that samples, like people respond and doesn't respond to antidepressants? That'll be so cool to look at, because like we could use this library to see their metabolites, and we could see if it's like different level of either like endogenous metabolite or gut microbial metabolite that's causing the changes in the response to the antidepressants.
So,
right. And
Scott,
now I'm done, Ron.
You may not know this, but we actually do have that data, not on everybody, but on more recently seen people, because we implemented a new questionnaire that's designed to identify antidepressant treatment responsiveness. So what Scott was referring to is this idea of treatment-resistant depression that perhaps is driven by things like inflammation in the setting of HIV. So anyway, I just wanted to point out that we would have that information on some participants.
That'll be very interesting to look at. Actually, another collaborator we were having, Sarkis Mathmanian at Caltech, they recently noticed that when they feed the antidepressants to a mouse model, and if you give antibiotics to them at the same time, basically, if you have germ-free versus regular mice, their response to antidepressants was very different. So it's showing that gut microbiome metabolism was causing something to the treatment response to antidepressants. So that would be really interesting to look at if we could use the HNRC cohort and maybe validate or like see different patterns compared with the mouse model.
Great. And you just hinted at what was my question, which is systemic drug exposure and how it relates to gut drug concentrations, because presumably the drugs and their metabolites have to be absorbed from the gut in order to have an effect on the host, unless there's some other signaling mechanism like through the vagus nerve or something like that. But in any case, the context here is that there are many more enzymes that are encoded by microbial genes than that are encoded by the human genome. And this idea of the microbiome's impact on drug metabolism, as opposed to people's, genetics and other factors, the microbial contribution might be even greater than the host contribution to drug metabolism. And so this provides an opportunity for future development of precision medicine approaches.
Indeed, exactly. Thanks, Ron, for pointing that out.
And I think Takashi has a question. Oh, what about the drug exposure? Do we know how much exposure in the bloodstream is related to the gut microbiome, metabolite and drug concentrations, or I guess I should say the stool concentrations?
Are you asking if we could study the pharmacodynamics and pharmacokinetics using this resource?
Yeah, or just do the same patient samples paired in the fecal matter and in the blood to see how closely or not they match, recognizing that the gut barrier is disturbed in HIV, so there may be more entry of drugs from the gut lumen into the systemic circulation.
That's a very interesting thought. As you said, like the gut barrier was damaged with the HIV infection, we could do a comparison between people with and without HIV and see how the drug distribution changes, especially for like antidepressants that's commonly used by the population.
With apologies to Dr. Tsukamoto for speaking up, we do have a pharmacokinetics project. I don't know, but I think we do collect stool, and I don't know, but we could find out whether we've sent you those stools. But that would be one way to do it, would be to look at, we're already measuring antiretroviral concentrations and cannabis concentrations in blood and CSF in those people. And we could send you the stool and we could look to see what the relationship is between stool concentrations and blood pharmacokinetics.
That sounds good. One thing is, I think one thing I need to point out here is when we do this screening, without the chemical standards, we will not be able to quantify. So if you were able to provide us a stool sample, if you could also give us the drugs, like the standards for the drug they will be using, we will be able to provide the concentration data.
We'd probably
Let's talk about that more. Yeah.
Purchase them from the I forget what it's called, but it's a drug repository that because if you need pure samples of the drugs, the medications, then I don't know that we could actually get them from the patients themselves.
Oh, no, just the standards. Yeah, just the standard. Because I was
Yeah.
like Dr. Latendre, if I pronounce it correctly.
It's almost people. I was thinking when Dr. Ellis was asking you about Joe, if I get a lot of questions about that too. So people just say Dr. Scott.
Okay, if you were happy with that.
Or Scott,
So,
or Scott.
okay, because Dr. Scott mentioned that they already have like quantification, like concentration data on the blood and the CSF. So I'm thinking maybe they already have the chemical standard and that we could already use it in our quantification as well.
So Takashi, you've been very
Is
patient.
this my turn?
Okay. Your turn.
Sorry
Great.
about that.
No problem. Very nice talk, by the way. I'm a chemist, so very excited when you see a presentation with every slides filled with chemical structures. So two questions. I think one might be related to some of the discussion you had. So I guess in this case, for each analyte, you are annotating either it's yes or no, whether that drug exists or not. So you don't really have any quantitative information in it. But where you put the threshold for this, like it's a big decision to make for each analyte. And I'm sure there's some noise versus signal issues associated with that kind of analysis. The second question is, how do you define metabolite versus analogs? Because to me, in a broader definition, they all look like metabolites. So I just wanted to get the clarification on the definition you use for some of the slides you see those terms. Thank you.
Yeah, definitely. These are a great question, and thanks for giving me the chance to clarify all of them.
Yeah.
So regarding the quantification, as we like I was asking Dr. Scott exactly for the chemical standard because we cannot quantify, as you pointed out.
Right.
When we analyze these samples using untargeted mass spectrometry, we get peak area, which gives us relative abundance information. Like you could use the peak area to compare the response between samples, but that doesn't mean anything. That's not related to absolute concentration at all. So as you can see on this heat map, I basically plotted the drug response or drug peak area I observed in different samples from like the blue color means lower peak area, the red color means higher peak area. But that's all relative quantification. That's not absolute quantification. I cannot tell you the concentration of these drugs without chemical standards. So as you mentioned, there's a lot of noise happening in mass spectrometry for sure. So that's really like we need to decide it on based on the data we analyzed that day, and we need to go into the blank sample and see what's the threshold of the instrument that day. You can see on this plot here that at the e to the 4, the color is still blue, so that's the threshold I set for this data set.
Oh, okay, I got it. Yeah.
And when it comes to the drug metabolites and drug analog, so I really use this term to differentiate. So drug metabolites, I'm referring to, give me a second. I'm referring to the drug metabolites that I collected from the public library, which basically means we synthesize that drug metabolite. We NMR it, we know its structure, we know it's a metabolite of drug. We were really confident to connect this structure to this MS2 spectra. So for those entries, I call them metabolites because I'm really confident they were known drug metabolites. And then for the drug analogs, I am referring to the signal that I collected by searching the public data sets using our computational strategy. So the signals I collected this way, it's definitely signals related to the drug, but we were actually not sure if it's a drug metabolite, or it could be an impurity or transformation products, or it could even be some artifacts during the instrument analysis. So then to differentiate that less confident computationally derived drug relevant signal, we gave them the name drug analogs.
Oh, I see. So for like in this example, you see some of the fragments showing -14, which indicate it's a demethylation.
Exactly.
Right,
yes.
right. But you can not still call this as a metabolite because it's not identified as a metabolite in the database yet.
Exactly.
Okay,
It's
got it.
signal relevant or related to a drug. It's not
Yeah.
necessarily a host or microbial metabolites. Yeah, that's
Okay.
exactly why we call them drug analogs.
Right, but potentially those could be also metabolite.
It
Or
could be
most
a
likely.
metabolite.
Yeah. Okay, thank you.
Exactly.
Yep.
Exactly. Thanks for letting me clarify that.
Thank you.
Any others? Speak up in case I don't see your hand raised. Raha, I don't want to put you on the spot, but you had a comment in the chat, and I know you've been working with some of our data on drugs and who takes them and who doesn't, and how they're related to the comorbidities and to cognitive function and depression. Anything you want to add, any sort of areas of synergy between what you've been doing and what Nina's working on?
I just wanted to say this was a huge amount of work to compile all of this. Like we've tried to make an annotated library, and we have one, not drug specific, but this I already looked up the preprints and the GitHub repository. It's huge. There's so much in there. So I really appreciate you putting this together and making it public for us to access. So I'm going to see how we can tap into this. I've only gone through part of the files so far. I might email you. Because what we have is the name and then the relative concentration, and it looks like some of your databases are based on the Spectra, but I see that you have the opposite as well for the HNRC data that you loaded. Is that the same as HDRC in the GitHub? I'll put it in the chat. Just make sure we're looking at the same thing.
Okay. Sorry, Dr. Raha, I didn't actually quite get the last question. What do you mean by HDRC? But here's my email, and thanks for definitely looking into the resource. If you have any bugs or problem when you were using it, definitely reach out to me.
Okay, yeah, I'll get in touch. I think it was just a typo because the file is labeled HDRC quant, but it's in the HNRC folder. So I think it's a typo. I just wanted to make sure.
I'm actually not sure which one it is. You can email me and show me it. Yeah.
Okay,
later.
perfect. Thanks.
Any others? Okay. Well, if there are no other questions, thanks very much, Nina, and thanks to the generous number of participants who joined to hear the presentation.
Thank you all for the great feedback.
Thank you both. I appreciate it. Sorry, Nina. Yeah, thank you. And then just so everyone knows, next week is Carlo Colantuoni, and he's from Johns Hopkins, and he is speaking on harnessing public multiomics data to discover shared mechanisms in neuroinflammation and neurodegeneration.
All right, thanks.
Thanks, everybody. Thanks, Nina. Thanks, Ron.
Thank you all. Thanks, Ron. Thanks, Caroline.
Thank you, Nina.
Thank you.